So nobody wanted to introduce me, so I'm still the same guy as before. So I like it better like this. So in the last few months, we have been discussing with our funders, the Bill and Melinda Gates Foundation, but also with some of you, we've been discussing the type of activities that GenRe-Mekong is likely to conduct over the next period. So, we've had so far two phases of GenRe-Mekong. The first one where we develop a platform and we work with research projects to sort of make sense of the data. And now in the second phase, we work with the control programs to deliver information and so on. So the third phase is the bit where you guys are eliminating and we support as sort of accurately and rapidly as possible these efforts with genetic information that is relevant to the specific events that are happening. So you can think of it like in phase 2, we're more like surveying the territory. In phase 3, it's more like a sort of very much targeted analysis of the events. So one of the main aspects that we're dealing with is to support the countries where we operate in sort of taking charge of some of the things that until now we've been doing, whether it is analysis tasks, and as you know, we've been working on transferring the knowledge on analysis and so on for the last couple of years. We've had some training courses and we've tried to develop tools and all this. But also there is the question of sample processing. Right now, we have an excellent regional laboratory run by Dr. Nguyen where you guys route the samples from the countries. But of course, that's not the only way to do things. And in a situation where you need as rapid response as possible, some of the countries have expressed a desire to actually conduct some of these analyses within the country. It's always quite a difficult question. How much technology is it reasonable to implement within the country? How much technology is it good to outsource? How much technology do you really want to invest in? So there are many aspects that one must look at. And what I've done in the next few slides, I've tried to capture some of the different layers of technology adoption by the countries. And sometimes when we have conversations, especially at big meetings with the funders and so on, it boils down to very simple questions like, do they have an Illumina sequencer, right? And actually, that's whether the answer is yes or no, it almost says nothing about whether it makes any sense that the sequencing is done within the country. You have to look at a lot of different aspects. And I think we see clearly that there is scope for certain technology transfer, and there is scope for a certain amount of aggregation of resources and sort of regional processing. So I'd like to just go through some of these some of these thoughts. So let's explore some of the factors that actually affect whether or not we decide to implement a particular technology within the country. By the way, the one that's missing here, that is the most obvious one, it should be the first one, is whether you want to. You know, I mean, to be honest, nowadays, if somebody were to ask me, what, do you want to implement a whole genome sequencer and whatever? I'm sort of, well, or else I could just send my sample to BGI and get it sequenced, you know, because certain things are just quite complicated and you can outsource them. So the first question is really, do you want to? But let's assume that you want to. And so the question is, do you actually have the infrastructure and the specialized manpower or manpower that can be trained? And then what question is the technology trying to address? So obviously, the technology needed to do a PCR to test what species sample you've collected is very different from trying to establish whether a sample is imported or not. They are different questions that require very different levels of data analysis, different resolution of data. Another question is, how many samples do you have? Because if you're processing 1,000 samples or one sample, your economic bottom line will be very different. So cost efficiency, of course, is a major issue. And then several countries, of course, have regulatory limitations, like can they send samples abroad or not? Can they extract DNA and send that? And ultimately, the magic word in this conversation is always sustainability. Moving forward, is this technology transfer useful to you? And can it be maintained? And actually, if the funders were to walk away, would your country be able to find the money to do it over time? And sometimes, often in fact, that depends on whether this know-how is also reusable in other contexts. So, of course, it's not enough to say, oh, we have a sequencer. But if you have a sequencer, you could use it for a number of things, not just for malaria, and so that would be an investment for other things. So when we look at spot malaria, you know, the technology that GenRe is using, what does it mean to implement this in country? So what does it mean to transfer the capability that Dr. Nguyen has established in her laboratory in Ho Chi Minh City? So in essence, okay, there is the laboratory infrastructure that you need. You need PCR machines, you need an Illumina sequencer that we've, as we said. It is a fairly complex protocol. It requires quite extensive training. And by the way, if you want to do falciparum and Vivax, they're two separate protocols, so you kind of do things twice. And that's just the laboratory part, okay, which is already quite a high degree of complexity. Once you run your sequencer, you end up with a very, very large data file. And then the fun begins, because going from that to the data that you can analyze to make those nice little maps beforehand, there are actually many, many steps of informatics that you have to do. So you have to be able to manage the informatics data. You have to be able to process it. And by the way, right now, with GenRe, you're all contributing data, and we are actually putting it together. We have, by the way, we have very advanced data sharing in this project. The rest of the world is admiring us for this, but it doesn't come for free. It has to, if you do it all separately and you still want to know how your data fits into the context of your neighboring countries, the data sharing has to happen. So there are many things that have to be done in order to put together genetic report cards for spot malaria. And once we've done it, then you can address a whole bunch of use cases, which we said earlier on. But now think about this. If you set up to do spot malaria in the lab, basically, if you want to analyze 100 samples, that's great. You take a 96 well plate and you can analyze 100 samples. You can analyze 400 samples with a larger plate. But if you have only one sample, that's going to be not so great, because if you run it through the machine, it's going to cost you about 100 times more than it would cost you if you had 100 samples, if you know what I mean. So when you have sort of a regional resource and you're sharing it, you also have economy of scale. If you have half a dozen samples in each country, you put them together, they become a couple of dozen samples, then it's more economically viable to process. So you have to consider all of these things. So sample volume is actually one of the considerations. And cost efficiency, so it's not just the number of samples, it's also a regional lab that processes more samples can probably get the reagents at a better price and so on. But on the other hand, you have cost inefficiencies. Like if you have a regional lab, then you got to ship the samples that you collect and so on. So you got to balance all these things. And finally, we're still talking about implementing spot malaria within the country. There are other considerations, like when you send samples abroad, and you're already having to do this, you typically have to get permissions. You have to fulfill the Nagoya Protocol that control the sample movement between countries. If you implement it within your own organization, you'll actually have to think about some ethics. For example, do you need, depending on how you're funded, do you need to make the data ultimately public in a public repository? And if you do you need to remove any human DNA from it? So again, complexities of the bioinformatics. These are all things that right now we are actually doing within GenRe-Mekong. So as you move forward, a major thing to consider, and this is where over the next few months, when we come and visit you and ask you about your operations, that's where you can advise us. We keep on being asked, okay, how can GenRe-Mekong be reused for other types of problems? Can it be implemented as, I think the expression is, as a horizontal system, right? Not just for malaria, because, hey, malaria is going to go away. That's what we are trying to do, right? So if you set up a system like that, does that mean that when the target elimination data comes, you throw everything away? Or does it mean that, in fact, you have people who are trained to do a certain class of analysis tasks, a laboratory that's equipped for doing certain types of analysis tasks, and so on? So I think at this level, at the level where we decide whether the technology transfer is a good idea or not, I think we need to start thinking about these things. What is the sustainability beyond the current moment? So, by the way, I'm not pretending that these are simple questions. These are difficult questions, and we need your help to tackle them. Now, in the previous slides, I talked about transferring the sequence, the amplicon sequencing technology, the spot malaria, which is what we normally use for the surveillance. But actually, there are probably other things that you could be doing at a local level, which are not perhaps as demanding in terms of resources, but are also very useful. So we mentioned earlier on, identifying a species. If you can do that with relatively straightforward qPCR analysis, then that's a very reasonable thing to want to implement in the country. So it's like a first level of surveillance, if you like. So different type of epidemiological questions basically require different levels of technology, different resolution. By resolution, what do we mean? It means how much data can you squeeze out of a sample, right? So, if you test an individual mutation, that's probably one bit of information that you're squeezing out, yes or no. If you are running the amplicon sequencing that GenRe-Mekong runs, you're getting kilobytes of information out, and so on and so forth. So you can do PCR. For instance, if you wanted to know what Kelch mutations you have, you could actually do nested PCR and send the product to be sequenced, or you might have a Sanger sequencer lying around. So that's kind of a next level of complexity, but also will produce very useful results, because if you're interested in artemisinin resistance, that's what you want to look for. Amplicon sequence is the next level of resolution. Then you have 100 SNPs plus tens of drug resistance mutations. So that's a next level of resolution. Then you can go for whole genome sequencing. That's basically, in the case of Plasmodium falciparum, it's about 30 million data points. Well, you don't really have 30 million data points. You usually just look at the ones that change, but it's still hundreds of thousands of data points, very high resolution. And it is very, very informative. You can get very, very accurate depictions of the epidemiology and so on, but they require a lot of resources to analyze, and a lot of specialization, and a lot of know-how. And, in all this time, I still haven't quite learned how to do it. So you have to make very big investments to be able to analyze this data. And that's why the amplicon sequencing that we deliver with GenRe-Mekong, that's a sort of more approachable level of analysis. And by the way, if you want to then analyze like amplifications, like you want to do MDR1 amplification and plus MEPS and so on, which are related to drug resistance, that's hard to do even with whole genome sequencing. So now we have technologies like nanopore technology with long reads, and you can use that to do copy number variations. But again, that's something that you could do instead with qPCR. So very often we have different mixes of technologies that can answer the questions that you are interested in. So, and of course, as your complexity goes up, as your resolution goes up, the costs tend to go up as well, as you might imagine. So, and here we're just showing if you're implementing the GenRe-Mekong protocol, the spot malaria protocol, you will have many, many steps that you need to execute. Not all of them in the lab. Some of them are online data analysis and so on. So there's a lot that has to be put in place. For whole genome sequencing, you end up having to manage to do alignments of millions and millions of sequencing reads, and then sort of detect changes and so on. It's all feasible, but it requires substantial resources, basically. And whether or not it is something that you want to do really depends on how much you think you're going to get out of this in the longer run. By the way, with whole genome sequencing, you have millions and millions of reads to align, and that's just for one sample. Then if you have 1,000 samples, you have to bring all this data together. So it quite rapidly explodes to a level of data management that requires a lot of management. Now, I mentioned Oxford Nanopore beforehand. This is a technology that has won a lot of fame in endemic countries because hey, you can have a sequencer that you can run from your laptop, and you can take it with you on a jeep and go and find somebody with malaria and sequence their genome and so on. And that sounds very attractive, except for the fact that if you really want to do this to scale and you have a lot of samples and you want to analyze them, this very quickly becomes a system that's just as demanding as having an Illumina sequencer ultimately. By the way, this is a setup that our colleagues from Malaria Gen did in Ghana in a field laboratory, and it does produce good data. You can do this, but you have to be realistic. If you want to do just a proof of concept and run it 10 times, it's easy. If you want to make this into a real viable long-term system, you have to put a lot more investment into this. So yeah, I mentioned if you want to do copy number variation, like long reads, the thing with Nanopore is it can read very long stretches of DNA, and therefore it is good, you know, if you want to look for things like gene duplications and so on. So it does have some sort of substantial advantages, and you know, it's something that's definitely worth looking at. But ultimately, you still always left with the issue of having people who can process the data. And this is really where things get complicated for a lot of organizations if they don't have a background in bioinformatics. So actually, what I think is a possible approach to this is that actually GenRe-Mekong, if you decide to go towards technology adoptions for specific type of tasks, the way we can support you is by providing online tools that support your data analysis and your data management. And again, I think over the next few months we'll have to understand what the actual needs are. But I think it is possible that, for example, well, in the same way that Anto showed earlier on that, he wants to get FASTQ files loaded up and tell you where that sample comes from. Similarly, to support in-country operations with relatively easy-to-use online tools, I think would be one way forward that would simplify the informatics problem for the countries. Actually, I had some slides on the various use cases here, and I'm not going to go through them because we have pretty much covered this in earlier presentations. But I just wanted to point out that, for example, we talked about identifying imported cases. Now, you might think, okay, that's a simple use case to describe, right? Is this an imported sample? But in fact, people don't actually ask that question. They ask, is this sample from here? Or is this sample coming from Africa? Okay, so the questions tend to be somewhat different. There's not a generic question. They are quite specialized questions. And there are, in fact, different tools that you can use to answer these different questions. For example, we have, if you want to test whether a sample came from Africa, actually, with the GenRe-Mekong barcodes, you get 99.4% accuracy just by looking at the barcode. Very simple machine learning algorithm. But actually, this paper's just been published a while back. You just need to qPCR 2 SNPs, and you can tell whether it's an African sample or not. So, and this, of course, you can implement in country. It's not that complex. So, so at this level, it's quite straightforward to implement the imported use case. However, if you want to test whether a sample came from Vietnam or Laos or Cambodia, then the question becomes quite different. And it's much, the machine learning algorithms are much less accurate, about 80% accuracy with our barcode. And you get more traction if you use whole genome sequencing, because you have just a lot more data. But you're still not going to get 100% accuracy. Why? Because a lot of samples, a lot of strains go over the border anyway, so your accuracy is never going to be 100%. And I think Anto has shown that quite well earlier on. Okay, and with that, I just wanted to mention something else, which is that earlier on we showed that we can actually reconstruct the ancestry of some of the circulating strains. And we've been able to do that in the case of an outbreak, to show that these actually were derived from strains that were circulating 15, 20 years ago. And the way we've been able to do that is by using whole genome sequencing. If you use whole genome sequencing, you have not one single variant or even a microhaplotype, just 2 or 3 variants. You have a lot of variants next to each other. And when you see that these variants are the same across many samples, then you know this cannot be a coincidence. They must have all acquired it from the same source, so you know they're related. Okay, whereas normally your variants are supposed to look a bit of this and a bit of that because parasites recombine with each other. So in this way, we've been able to reconstruct that if you take 2 strains, which we called KH3 and KH2, and we analyzed this sample that was from an outbreak in Laos in 2019, we could actually see that different parts of the genome were inherited either from one strain or from the other strain. And it looks like a mosaic of those gold and green tiles there. So with high resolution data, you can answer a question in a very sophisticated manner. But the price you pay is the complexity in data management and analysis and so on. There's no free lunch. Well, actually there was today, but yeah. Okay, and with this, just to recap, deciding whether or not you're going to implement a technology in country is a choice that must be made considering many factors. Complexity is one, cost is another one, maintainability and sustainability is another one. There's no one size fits all, and I think it's very important to consider what the impact of implementing the technology will be on other systems that you may have in place in your country. And with this, thank you very much. And I can take questions if you have any. Yes, Sarah.
Yeah, thanks for a lovely summary, Oliver. I was just thinking on the Oxford nanopore technologies, as Anto mentioned earlier, one consideration is whether or not you can wash the flow cell if we're thinking about, when you just have a few samples versus a lot of samples. Also, I just wondered if you had any information, because if you're using this just to target certain markers, right, rather than doing whole genome, and you just want to look at the parasite, you're probably still going to have to do some form of amplification, right? If you use ONT. So then are you still going to get the length, right? If you still have to do a pre-amplification first to get your specific targets, you'll still have some of those challenges with length. And also, just from our experiences of working with ONT, where we have tried to get long fragments of, say, 6 kb, if you're using things like dried blood spots, that might not work so well. It might be a bit more fragmented. So just, I don't know if that's more of a comment or a question, but yeah, just some food for thought.
I think to summarize what Sarah just said, she said that I only told you a little bit of the story. Yeah, yeah. No, everything you said is very true. Your ability to get very long reads and looking at copy numbers, etc., may go away if you have a sample with very little DNA, for example, such a blood spot, right? So it may be very different if you're able to either culture or take from venous blood or something. And if you need to do amplification, which is what we do in our spot malaria platform, then the amplification typically works on smaller segments, not small, but smaller than than you might want for your long sequencing. So it is true there are limitations. Certainly, I mean, Sarah, of course, remembers very well because she was deeply involved in this. But 15 years ago, it was impossible to sequence from a blood spot, to sequence falciparum from a blood spot. We couldn't do it because you had to. The only way to get enough DNA was to filter out the human DNA, and you had to put it through syringes and things like that. And there was just not enough DNA in a blood spot. So we had to develop amplification methods, but amplification methods come at a cost. You have to break down the DNA and all this type of stuff. So without going into this any further, it's all true. The advantages of the Oxford Nanopore have some limitations, but you still get longer reads than you would from Illumina, which is still useful, I reckon. Was there something else you had said? Yes, the number of samples.
Yeah, so just to clarify on the longer reads, I guess you would get longer reads if you're sequencing your entire sample, right? And you're not doing what we do with GenRe-Mekong, where you're doing, you've got PCRs, right, targeting specific regions of interest. But if you're going to sequence your entire sample, then of course you're going to get a lot of human DNA in there, right? So you're going to get a lovely human genome. So I think those are just things to consider.
And you may still not be able to do it from a blood spot.
And you might not, exactly,
yeah,
because of the fragmentation of things.
So, everything has a trade-off. And I really literally skimmed over the surface on these slides. Yes.
Have a quick question, and just in terms of some of the practicalities. So if the countries were going to adopt this, would they be expected to do both the lab work and the bioinformatics analytics, or could they do the sequencing and then the like the analytics part of it could be done elsewhere, or would they be expected to do?
I think the part where we can support the country implementation in multiple ways. One, for instance, Nguyen and her group have immense laboratory expertise, and they can transfer that knowledge, they can handhold, they can do the laboratory implementation, so that should work. And my team, on the other hand, is more on the informatics side and so on. And what we would look to do is to provide support such that that burden isn't on the local implementation. And one way to do that is to provide online tools such that they could upload their data and somehow we process it. So, or we would have, we would maybe manage some data processing pipelines that they could also access. I haven't given this enough thought to give you a working model, but the idea would be that no, they don't have to set up a big bioinformatics group, because that would be too onerous, really. Unless, of course, that's really what they want to do, in which case we support them do that. Make sense?
Yeah.
Thank you. Martin?
Yeah, thank you very much. So my thinking is that with the development of the lab analysis technology as well as artificial intelligence development, it should be more easy or as well as cheap to do some to build the lab analysis network is very possible. So my thinking is that because toward the last mile to elimination as well as post-elimination stages, different countries, different area may face different challenges, different issues, different. So should we consider the short term or long term, the issue, what we try to solve, through this innovative technology, through this innovative platform, such as in China. Currently, we want to understand more the risk of local transmission from imported case, as well as the border malaria cases. Because if we put a lot of resources, do a lot of capacity buildings through the national program to build this platform, we hope to have long-term supporting to use these technologies for the future, not only for the next 3 or 5 years, even the 10 or more years, to consolidate the malaria elimination efforts and to support the post-elimination efforts.
So you're asking what
What's the
technology
priority?
does it make sense to adopt
Sure,
and to
sure.
invest in?
Sure.
Yeah?
Yeah.
Yeah, that's $1 million question, I think. Well, I mean, and I suspect different countries will have different circumstances that will make different amounts of sense to them. I think, I mean, yeah, off the top of my head, I really couldn't answer this, and I think it really takes some serious thinking and analyzing in the context of each individual country. What I do know is that when I ask this sort of question, if I'm talking to the funders, they actually say, well, try to imagine that in a few years' time the funding will not be there. And which is I think their definition of sustainability, right? Today you have me, tomorrow you might not have me. So what made sense that you spend the money on, so that you're not crying when the time comes. But this is very high level comment, but I think then you need to look at the details in each individual country to really know what the answer is to that. Yeah, I think the sense of my presentation was to say, don't believe anybody who tells you, this is very simple. It doesn't require that you do anything. No, it's difficult stuff. It requires investment, and you got to see whether it's worth doing. And it may well be really worth doing, but it does require an analysis. It was kind of not really an answer, but Please.
Yeah, I think very much along the same lines. I guess the question about sort of whether it's worth setting up in-country sort of facilities like this is also, and I'd be interested in hearing what's the vision for, doing vector screening panels. Is there a drive to actually think about sort of diversifying across disease systems? Do the country partners want to put in, AMR screening or dengue virus screening? So thinking beyond a single disease system. So I think having that vision piece would be quite useful. And in phase 3 of GenRe, is there a view of doing sort of vector work, or is it just sort of focused on Plasmodium, Falciparum, and Vivax? Or does it even extend into Knowles eye? Are we going to start, or you going to start thinking about the non-human primate malarias as well?
No, that's an excellent point. And actually, that's a very good example of what they mean when they're talking about horizontal systems. Like the vector systems would be a very good example of that. As far as to whether GenRe in phase 3 is planning to do this, actually, it's a sort of it's something that came up quite recently, but it does seem like a direction that we should follow very seriously. Setting up some vector type of monitoring. Do you want to comment on that, Nian? Not yet, but we are seriously thinking about it because this is feedback that we got from the control programs. There is a lot of interest. They would like this data. we need to figure out what is a good entry point into it. Thank you.
Yeah, actually, just reflect to what Olivo has shared about the futures of surveillance and the application of molecular. Actually, we do a survey with the National Malaria Control Program today on the feasibility of implement the procedure in country, and actually from we have various of the feedback from the NMCP, but actually I think most of them prefer to from the answer, most of them prefer to implement the protocol within the NMCP lab. Some institutions prefer to send to the central lab because they will save the cost of human resource to doing this. But in general, I thought that all of them want to learn more with the technique and the bioinformatics tool. Then that will be the very highlight point to move forward with the next phase on transferring the technique and also the analysis tool. And we will, based on the information collected today, for moving forward with further in-depth discussion with each country, maybe in the coming years, for how can we do support of transferring the technique, and about your capacity to doing this, or for any further support you need from GenRe-Mekong in general. But it is not sufficient time today for moving deep with the discussion. But actually, we got the feedback from you on the in-country implementation, and we hope that will be the discussion in the new years. And there are also several points that concern related to the training for human resource for the NMCP, and also the concern about the maintain the technique for future, not only for use with malaria but for other disease like Martin and other has comment, and we note that, and it is from one of the points we want to transfer the technique to support you to use it for the future. Thank you.